All right, let's dive into something that sounds like it's straight out of science fiction, but I promise you, it's real. We're going to talk about how a team of scientists took one of the world's very first movies and stored it, piece by piece, inside the DNA of living bacteria. Yeah, you heard that right.
So let's set the stage. What you're looking at is Edward Muybridge's iconic galloping horse from way back in the 1870s. This was a huge deal. It was this series of photos that finally proved that all four of a horse's hooves are off the ground at the same time when it gallops. It's basically one of the first motion pictures ever.
Now fast forward about 150 years. In 2017, a team at Harvard, led by the famous geneticist George Church, took 5 frames from that exact movie and encoded them directly into the genetic code of living, multiplying E. coli bacteria.
Which of course leads us to the big question, the one we're going to unpack in this explainer. How is that even possible? How do you actually turn a living cell into a hard drive? How do you take something digital, like a movie, and write it into DNA? Well, let's break it down.
To really get how they pulled this off, we need to understand the 2 main tools they used. And the coolest part is, they didn't have to invent them. They just had to figure out how to use the incredible tools that nature already created.
So first off, why even bother using DNA for storage? Well, if you put it side by side with a regular hard drive, the difference is just staggering. Our drives might last a few decades if we're lucky. DNA, it can hold on to information for thousands of years. But the real mindbender is the density. DNA is an unbelievably compact way to store information, way more efficient than anything we've ever built.
The second tool is CRISPR. Now, I know you've probably heard of CRISPR as this revolutionary gene editing tool, but that's us hijacking it. Its day job in bacteria is actually to be an immune system. When a virus attacks, these little proteins, named Cas1 and Cas2, snatch a piece of the virus's DNA and they stitch it right into the bacteria's own genome. It's like a molecular most wanted poster. It lets the bacteria remember the threat for the future. So you see, it's a natural data recorder.
So, with those 2 pieces in place, DNA as the paper and CRISPR as the pen, the scientists decided to do a test run. Before trying to encode a whole movie, they started with something a little simpler, a static image of a human hand.
So here's the basic game plan. First, they had to translate the image into a language the bacteria could understand. They converted the color of every single pixel into a unique DNA code. Second, they fed all these little DNA snippets to a huge population of E. coli. Third, they gave the bacteria some time to grow and incorporate that DNA. Then they sequenced it all to read what was stored. And finally, they used a computer to decode that DNA sequence and put the picture back together. And what happened? It worked. It worked beautifully. On the left, you've got the original picture. And on the right, that's the version they rebuilt completely from the DNA of those bacteria. The accuracy is what's so amazing. They got back 96% of the pixel data correctly. They'd actually stored and retrieved a complex image from a living organism. But getting to that 96% wasn't exactly a walk in the park. They had to do some troubleshooting. They had to figure out the best way to talk to the CRISPR system. It was kind of like they were learning a new programming language, the language of bacterial data storage.
At first, they tried 2 different approaches. One was a rigid strategy, where one DNA letter, A, T, C, or G, stood for one of 4 colors. Simple. The other was a flexible strategy. This one was more complex. It used a group of 3 DNA bases to code for one of 21 different colors. And as you can see, the flexible one was the clear winner. The accuracy jumped from around 88% all the way up to that 96% mark.
So, why was it so much better? Well, this chart shows something called acquisition frequency, which is just a fancy way of saying how good each strategy was at getting the bacteria to actually grab and save the DNA. The flexible strategy gave the scientists more room to design DNA sequences that the bacteria's natural machinery just liked better. It made the whole process way more efficient. And by figuring that out, they basically uncovered a rulebook for writing good DNA data. They found that CRISPR works best if you keep the amount of G and C letters balanced at around 50%. It also doesn't like long, stuttering repeats of the same letter, and you have to get rid of certain internal codes that can confuse the system. But the biggest breakthrough they had was discovering what was basically a hidden password, a tiny, specific sequence at the end of the DNA snippet that acted like a secret handshake with the CRISPR system. The researchers put it pretty clearly in the paper, saying, the final 3 nucleotides determined acquisition frequency. What does that mean? It means a specific 3-letter code, TGA, was like a magic word that gave them super high storage rates. Another code, CCT, was a total dud. That little sequence was the key that unlocked the whole thing. It was like shouting, "Hey CRISPR, over here, record this one."
Okay, so now they're armed with all this new insight. They have the better flexible code, they have the rulebook for writing good data, and they've got the secret password. The team was finally ready to go for the main event, encoding the Galloping Horse movie. And the scale here was pretty impressive for this kind of work. They took 5 frames of that movie, with each frame being 36 by 26 pixels. All told, that came out to about 2.6 kilobytes of data. Not a lot for your laptop, but a massive amount to write into the genomes of living bacteria.
So how do you encode a sequence, a movie? You do it over time. It was a patient process. On day 1, they gave the bacteria the DNA for frame 1. On day 2, they gave that same population of bacteria the DNA for frame 2. And so on, for 5 straight days.
This brings up a really important question, right? How did they keep the frames in order? And the answer is just so elegant, because they didn't have to do anything special. CRISPR does it for them. It's a first-in, last-out system. It always adds new memories to the very front of its storage list, which pushes the older memories further down. So the order of the frames was kept perfectly just by the order they were introduced.
And here it is, the grand finale. By sequencing the DNA from all those bacteria and looking at the position of each frame's code, they reconstructed the movie. And the result? Over 90% accuracy, and the frame order was recalled perfectly. They did it. It actually worked.
So this is obviously one of the coolest things I've ever heard of. But the point wasn't just to make the world's tiniest, most biological movie theater. So why does all this actually matter? What's the bigger picture? Well, the researchers themselves summed it up perfectly. They said, this proves that the system can capture and stably store practical amounts of real data within the genomes of populations of living cells. In other words, they showed that using living things to record complex, time-stamped information isn't just a wild idea anymore. It's a reality.
And that is the real takeaway here. The ultimate goal isn't to replace your thumb drive with a test tube of E. coli. It's about creating living recorders. Think about it. Imagine you could engineer cells that can monitor their own environment, say from inside your body, and keep a diary of biological events over time, written directly into their own DNA.
So I'll just leave you with this final thought. If we can program a cell to record an old black and white movie, frame by frame, what could we one day program a cell to record from inside a developing brain, or from within a growing tumor? The possibilities are just incredible to think about.